right? So if you Google, I don't know, oranges, then the length of your query is 7, right, or 8 characters long, right? So if the time it took to search for something was only the length, order the length of your search query rather than the length of the universe of possible, pages where oranges occur, then that's fantastic, and that's exactly what this does, right? So essentially, and then there's a bunch of other cool algorithms, and I don't want to get into the algorithm. Well, I will get into it a little bit, and I can get into it at arbitrary detail if you like. But the point is that there's a number of cool algorithms that go into just telling you where this read actually belongs on the genome. And the first step in that is to do some kind of a global search. And then the second step in that is to do some kind of a local adjustment of what you found. And those two actually tell you exactly how a particular read aligns to a reference. Right? And in doing so, this is interesting, right? So essentially we have this read coming in from somewhere, and this is the reference. Remember, the reference is usually a healthy genome, right? It's a normal human genome, right? And this read actually comes in from some real sample. So some person has this. So what this is saying is that there's one insertion here of an A, right? And so there's a deletion here of a C. And then there's a mismatch here, A is turned into G. So this is a lot of information in and of itself, right? But this is exactly the kind of thing that you saw yesterday as well, right? Essentially saying, what mutations does this individual contain? The way to do that, the first step is alignment, right? And then the alignment problem is actually quite complex, and it's very, very computationally intensive. Right? It takes 90 hours on just a single thread. Right? With 30 threads, it might take about 9 to 10 hours. So BWA is like this, for instance. So can we use the GPU? That was the sort of motivating question. Right? So this is a very specialized piece of technology which has been used traditionally in the past to solve computer graphics type problems, repurposed now to solve hopefully a problem in in genomics, right? So essentially, what this is if you want to render a lot of if you want to render an image with a lot of pixels in it, then you have a lot of processors, each of which is doing some bit of the work, and usually that unit of work is actually the rendering of that pixel in the graphics world. Now you can use that, you can repurpose that, and make it massively parallel once again, but instead of rendering pixels on a screen, you're actually doing something related to genomics. Maybe. Right? That was the hope when we started off this project, right? And you can get really high speed ups of up to 500x depending on the problem, right? So and so what we did was we looked at how to speed up Smith-Waterman, which is as I said earlier, is the local alignment tool, right? This part, essentially saying once you do the global alignment and know roughly where it belongs, you do the local alignment to fiddle around with it and get the most optimal way to align the screen to that reference with the minimum number of changes, right? And so anyway, so the Smith-Waterman is actually quite challenging, to put on the GPU, and we spent some time doing this. Essentially, what this does is it takes these reads and the reference substrings, and it uses certain penalties for gaps and mismatches, and then the dynamic programming problem actually tells you, given these penalties and given this read and reference, what is the optimal score and what is the corresponding alignment, the alignment that corresponds to that optimal score, right? And ignore all of the technical stuff, I can go into it. But essentially, the big takeaway here is that these 3 steps, right, they're not particularly, amenable to being put on the GPU because they're all very text heavy, and they involve the manipulation of characters, which is traditionally not something you would do on a GPU. and there's also part of it was prior work, but part of it was, the hard part was not done at the time that we set out to do this. The big takeaway here is that you might, if you ever write any piece of code, you might start to get obsessed with this particular ratio. It's basically how many times do you access the memory for a given mathematical operation, right? So if you had a program that just reversed a string, then that doesn't do any mathematical operations at all. It's just string manipulation, right? But on the other hand, if you had something, maybe computing, the GCD or something, right? So that's very mathematically heavy. But most programs sort of lie within that, between very mathematically heavy and very memory heavy. Most programs lie in that continuum. And the more mathematically heavy something is, the more amenable it is to being sped up on the GPU. I think that's really the takeaway here. But essentially, this was the opposite. So that was one of the challenges that we had to face. And we came up with a parallelization scheme which was fairly simple, and then we spent a lot of time, figuring out exactly how. So this stuff, I think it's nice. It's a nice slide, but it really doesn't tell you a whole lot because there's a lot of detail. I think the important thing to take away here is that you have to spend a lot of time optimizing your code for the platform that you want to put your code on. And this is something that we don't normally do if we're coding in C++ for the computer, for the CPU. But as soon as you start to get into something like a GPU or an FPGA, you end up having to do things like this, right? And long story short, we had a 54x speed up, and we had some good statistics. We had an extremely fast, algorithm for Smith-Waterman on the GPU. We used multiple GPUs to, improve it further, etc., right? And then we had some good, we had a good alignment tool, right? And that was fantastic. But on the other hand, we had to do a bunch of other implementation challenges, which I won't get into here. The important thing to remember is that this couldn't get much better because of reasons that were beyond our control. Because the time it took to transfer information from the CPU to the GPU was nearly the time it took for the entire kernel to execute. So essentially, that was one of the big takeaways. The other big takeaway was this, and if you ever try to optimize a computer program, you will find that sometimes you're so good, or rather your program is so good that you start to wonder, can I read faster? That's a good headache to have. Can I write faster? Can I read faster? Right? Those are good headaches to have. Right? So essentially, these are the kinds of things. This is a fun graph that I drew where how much time does it take for an average programmer like himself to go from some rate of execution to some other rate of execution, right? So this is basically speed of that program, and this is how much time I spend on it, right? Hopefully this, I don't know if this may not be very flattering, I don't know, but the point is that it kind of starts to saturate after a point. So you're actually spending money, right? Because you're spending programmer time optimizing a program that is increasingly harder and harder to optimize. So at what point do you just say, hey, I'm going to stop, right? And hopefully if you know what you're doing, you know when to stop because it stops being useful after that point. So these are some interesting things to do. But we didn't stop there. We went ahead and optimized the entire workflow, and we could do the entire thing. If you recall, just alignment in the previous slide was about 9 hours. So we did the entire workflow. So there's a bunch of stuff here: alignment, quality recalibration, realignment, rank calling. In about 6 to 7 hours. So that was really nice. Right. So this is this is one way to look at the big data problem. So I notice I have 7 minutes. I think that's perfect. But really, I think Strand has sort of moved on from this paradigm of just looking at this is one aspect. This is the computational aspect often this is the least important aspect. That's what we figured out, right? Because, so, let me tell you something. Whereas we're not at the scale right now, and we don't expect to be at the scale where aligning a whole genome in 7 hours is a bottleneck. Because often the the processes in the laboratory and the processes of data transfer and storage and all of those other things are much, much slower. So this is not the bottleneck really. The bottleneck lies elsewhere. And so in investigating those bottlenecks, we've actually, we're also, and I do want to touch upon briefly on the kinds of work that we're doing now, which draws upon some of our experience with, big data in the computational realm and optimizing that, but, also hopefully sort of sends us on a path that's a little more diverse, right? So, this is, again, complete context switch here, so forgive me. But what I wanted to give you a flavor of was, there's a lot more to genomics, I think, than just what you saw over the past 20 minutes or so. That part is usually what is called secondary analysis, right? And secondary analysis is supposed to be easy because you set up pipelines to do it. So yesterday you saw alignment, assembly, etc. There were standardized pipelines to do that. What a bioinformatician typically does is they set up a pipeline where all the parameters are set, right? And that's validated in a lab, and that's it. And that doesn't change for a long time. That's secondary analysis. But once you do the secondary analysis, what Dr. Wall has been showing you over the past 2 days or so is actually the interesting part, is the tertiary analysis, right? So it's essentially saying after the computational automation is done, how do I actually interpret this data biologically? What does it mean? What do these numbers mean? So for instance, so this is secondary analysis for the first part, but essentially what is this study? This study, I mean, imagine that there is this thing called a small RNA. I don't want to get into that. Small RNA is sort of a regulatory RNA, which is, which has a certain impact in, certain kinds of processes in the cell. But essentially what we're trying to say here is, how do you make a process as complex as this somehow intuitive, right? And a lot of this is not secondary analysis. It's actually tertiary, and so people are actually having to look at this and so on. So just to give you a sense, right? So this is in a in an area called single cell RNA-seq. And it's a very, and I really recommend that you all, if you haven't heard of it, please, go and look it up, because it is definitely the next big thing. A lot of things are happening here. So essentially, we're analyzing these 539 cells with so many millions and millions of reads, right? So again, another big data problem. And the way we're doing that, in the end, what we're trying to do is to come up with a biological, with several biological insights. So checking for known markers is one of them, and hopefully that you've seen in the last 2 days. Trying to detect novel genes that weren't actually, perhaps known before, perhaps unique to these samples. That's another one of the goals. So again, very biological, right? And also, how are these cell types differentiated? So there are many cells, single cell RNA, how are these cell types differentiated, right? So all of this is in the work of tertiary analysis, right? And where tertiary analysis, and I'm going to quickly change the context once again, with your permission, to the last part, hopefully. And really, I think another thing that Dr. Wall also touched upon today, interestingly, and that's why I wanted to flash this a little bit, is ignore there's a lot of detail here, but I think the important thing to notice here is that there is a clinical element, and so these are two studies that we're doing. One in non-invasive oral cancer, and another one in ovarian cancer. And in each of these, there's a study design element, a clinical element, a sample processing element, and a molecular analysis element. Right? Sorry, should be analysis. Excuse me. So essentially, this starts to speak to the kind of integration we're talking about, because this is just one example of a study that we have. I think I wanted to talk to you briefly about it because it sort of integrates a lot of the themes that we've been looking at Strand for a while now. It's an oral cancer study we're doing with the University of Chicago. Essentially, what we're trying to say is, can saliva be used as a prognostic indicator for oral cancer? Oral cancer is a very big, problem in India. It's huge. The incidence is huge here, right? And the idea is that if you can get someone to spit, and if we could sequence that, and by sequencing that we could do an early detection, that would be fantastic, right? And of course, it has to be cost effective, etc., etc. But that's the goal of this study. But what I'm trying to draw your attention to is that this is massive in many, many different ways, right? Not just in the informatics way, but you actually have to have a good power analysis. You have to figure out, how many patients you need for the study, right? What are the stages in which you will recruit patients, right? What kinds of samples are you going to collect from them? How are you going to collect those samples? A lot of them are chewing, all the time, right? They're chewing betel nut, they're chewing tobacco, etc. Right? So how are you going to collect 5 minutes? How are you going to collect the sample from them? When are you going to collect the sample? Right? You want them to rinse them out because you don't want the residue from the leaves to be part of your. But at the same time, you don't want the cells to be washed away, the malignant cells if they exist. Right? So what's a good sample collection protocol? Right? So if you start to think about, so when you just look at a number of, a row of numbers, in a matrix, which is what RNA-seq is, or small cell, single cell or small RNA, etc. You're not aware of the dimensions of this, right? So the dimensions of this really extend well past, the logistics. There's molecular, there's lab work, there's convincing people to do it the right way, right? So, I think that's really, it's been a very humbling process for us because, especially for me, because I'm a software guy, I used to be, right? And when you actually start to get into the study, you start to see, the clinical study, you start to see that there's so much more you got to do. It's not enough to just know how to analyze RNA-seq, right? It's just not. And the other thing about the analysis, which is interesting, is that here it's actually maybe somewhat simple. Essentially, what we're doing is there's saliva, right? And then there's plasma, and then there's the solid tumor itself. So these patients that we're recruiting all have oral cancer. They have a tumor. They have, of course, saliva, and we're taking blood from them. And what we're trying to do is we're trying to look at the mutation profile in the solid tumor and seeing how well it matches with the plasma and saliva. So this is a three-way, concordance, right? It's just a triple well, you can think of it that way, if you like. But it's much more complex than that because it involves very, very careful and patient tuning of all of the elements to make sure. So that's the whole validation aspect, which I don't have time to get into here. But I think what we're trying to say is that the kinds of steps that are involved in a real world study and or a program like this extend far past big data. But somehow big data, the ability to do big data is very much part of, these kinds of studies. The ability to integrate, in fact, analysis from various different sources and say something meaningful about, what's happening I think that's really, and that's where we're at, and I think that's really interesting and incredibly challenging. So for instance, so this was a bioinformatics exercise, right? So the question we're asking here, and this is kind of, right, this is what is known as translational bioinformatics in some sense. We're asking, given, so what we're doing is, we had a certain budget for the study, right? And within that budget, we need to design an NGS panel, right? And the wider the NGS panel, the more it costs to sequence someone. But of course, the better the specificity. Of course, if you sequence the entire genome, then you catch everyone, right? So the optimization problem we're trying to solve here is we have a budget, that's the panel length. What subset of genes maximizes the number of patients you can catch with that panel, right? So that's an optimization problem. And I'm not sure if it's in any bioinformatics textbook. It's not very classical, but it's the kind of thing that you have to do routinely if you have a budget, right? So this was really cool. And so, we ended up doing this as I won't get into how we did the optimization, but essentially we developed this panel. This is MD Anderson, this is this is ICGC data from Dr. Majumdar's paper, and then this is TCGA. So essentially, this panel of, I think it's 8 genes is what we came up with, and so on. So these are some of the real challenges, and I don't want to get into this anymore. I did want to sort of leave this as the concluding thought here is that essentially there's a lot that goes on in bioinformatics is merely the terminal point of a very vast and complex sort of association of people from the clinic, people from the lab, people from computer science, and people from bioinformatics. And I think the more we start to appreciate that, I think the ability to appreciate that, I think, is really the enriching exercise, and also is a fantastic challenge. So, thank you so much for that.
So we are behind time, so we'll take the questions during dinner time. So I would like to thank you, sir, for such an interesting talk. I would request Dr. Graham to please felicitate. So now moving to the next talk, our next speaker is Dr. Ram. So he has done his PhD in ovarian cancer from ACTRAC and postdoctoral study in multiple myeloma from MD Anderson Cancer Center. He has co-authored in journals like Cell, Leukemia, and Blood. So please welcome Dr. Ram.
Thanks, Andy, for that introduction. And thanks, Professor Gilo, for allowing me to have my talk here. I did my PhD from ACTRAC, Harvard Navi must be known, and I was working on ovarian cancer, basically in cancer stem cells and chemoresistance. I know my talk will be slightly different from bioinformatics, so more of biology and less of informatics. MD Anderson Cancer Center is the world's largest center for cancer biology itself, and you can see us strike out here. So we have a focus of negating out the cancer from the world, and then have more of clinical aspect rather than the basic research. And you can see more in the background is just not for the beautification. So we have a moonshot program in MD Anderson. So these moonshot programs involves cohort of PIs with heavily funded group, and they work on patient samples and clinics to make more of higher survival, how they can have more clinical impact on the patients rather than just the basic research, and they can do whatever they can do in their capacity. So today my talk will be involving 17p deletion. So 17p is nothing but a chromosome which has a p arm and a q arm, and 17 is the location for the p arm. So that gets deleted. So once there is a 17p deletion, it makes a cell therapeutically vulnerable. So what I want to say is, say if a person is diabetic and he has one kidney working and one kidney is not working, right? So if you want to surgically kill him, you'll try to escalate the other kidney, right? So he's more vulnerable when he has only one kidney, right? So once we have a 17 deletion, that population becomes more vulnerable for the therapeutic aspect. So when I said multiple myeloma is plasma cell myeloma, and it's plasma cells is a normal cells in our body. Much excess amount is not good. So excessive number of abnormal plasma cells in the bone marrow, and it leads to the overproduction of intact monoclonal immunoglobulins. So you don't need to go into that details, but the basic idea is you have the amplifications of plasma cells, and these plasma cells can have more of decalcification. So bone is consistent of calcium, and when you decalcify the bone, your bone becomes more fragile. So that's the primary symptom of the multiple myeloma patient. When you have a fragile bone, it collapses. And the first aspect is the stem cell. When your stem cell can have T lymphocyte formation and your B lymphocytes. And this is a normal procedure in our body for the development. And you have antigens, activated T cells, plasma cells, and antibodies. And these antibodies take care of your immune system, right? But when this gets deregulated because of some genetic damage, this damaged B lymphocyte forms a malignant plasma cells. And these plasma cells create a lot of antibodies, which is not antigenic to some other microbes, but is more antigenic to its own body. So it is not good for your own body as well. So there are probable causes which can lead to multiple myeloma, but there is no concurrent study which can show that there is an exact mechanism of action where it leads to multiple myeloma. But there is a probable hypothesis that genetic causes is one of them, and environmental or occupational causes, and radiation might be one of them. So when they look at the survivors from Nagasaki bombing, in where 29 people died of the multiple myeloma from 1950 to 1976, which is just a speculation. But it says that radiation could be one of the major reasons which can drive multiple myeloma as well. The clinical feature for multiple myeloma is CRAB. And when I say CRAB, it means the calcium elevation. You have a renal failure, you have anemia, and you have more probable bone lesions. So when I say bone lesions, it's very painful, and you can see image over here is a scan of a hand, and you can see these punched out patterns. So these bones over here have been decalcified. So you can imagine a status of a patient running from the multiple myeloma is a fragile bone, and his neck and vertebrae all gets merged, and they have complaint of bone pain as well. And if you see this one, this is spinal cord, and when they have a multiple myeloma, they have inflammation in these regions, and these spinal cords are the first region which gets affected. And since they are carrying your spinal nerves, so the most prominent effect you see is the paralysis. So the patient having multiple myeloma leads to often more of the paralysis. Now coming to the disease background, we classify multiple myeloma in two groups. So one is a low risk and other is a high risk. As the name suggests, the low risk patient having multiple myeloma is treated very well and the recurrence is very less. But what we are interested in the high risk patients, including 17p deletion. So for these patients, the survival period is very less. And even if they are responding to the primary therapy against cortisone, which is a proteasome inhibitor, they respond well in the first site, but again they come up with a relapse. So the major issue is treating the patient with the high risk, not the low risk, because low risk has a standard treatment module. And this is a heat map for the copy gain and copy loss of 17p. So this whole span is your 17 chromosome, and this region allocates your 17p deletion. So blue line basically depicts your deletion region, and the red is your amplification. So the whole patient sample. So a single line is your single patient sample with 17 chromosome, and this is the P region of 17 chromosome which is getting deleted. So approximately 10 to 11% of the melanoma patients of high risk have 17p deletion, and rest of them have different kinds of amplifications and other deletions as well. And this data was analyzed from MMRC database, which is nothing but multiple myeloma research consortium, which is a publicly available database. And this is a heat map showing other alterations in myeloma patients. So you can see 1p 13.2 deletion. So that means chromosome number 1, arm P, location 13.2 is deleted in that particular patient, and which is significant and which leads to more high risk. Similarly, you have 1q22. So chromosome number 1, Q arm, and 22 position is deleted. So similarly, we figured out that 17p chromosome was important because 17p is having your p53 gene, which is a housekeeping, gene keeper for your tumor suppressor. So we got interested in 17p 13.2 because the 17p not only harbors your p53 and it also harbors your RNA polymerase 2, which is a major transcription machinery for transcriptional process. So the red region are the significantly amplified regions in the myeloma patients, and the blue region depicts your deletions. And when we look at the survival across the different patient samples, we found that the yellow line are the samples which has abnormal 17p, that is 17p deleted, and this blue line is your non-deleted, which is normal. So when we compare survival, these patients have a very low survival as compared to the patients which have wild type p53 or wild type 17p. That suggests that the 17p deletion is very important for the treatment module when we consider a high-risk patients. And when we figured out that the 17p often co-deletes your polar 2A. Polar 2A is nothing but polymerase 2A. And 7 to 11% of the newly diagnosed cases and 22% of relapse and refractory settings patients have 17p deletion. And PFS is your progression-free survival. So the survival time where there is no progression, right? So that is 2 years, and overall survival is 50%. And then we wanted to know what is the polymerase 2A probe intensity in 17p deployed, which is wild type, and 17p deleted. So we do see a significant difference in polymerase 2A mRNA expression. So not necessarily that copy number loss will ultimately lead to your low number of mRNA level as well. But here we do see the transcript level also goes down with loss of copy. So there are two situations. One, we have a low copy number, but the other copy can take over the low copy number loss, right? So the equivalent amount of mRNAs in the system will be equal, even if there is a loss of copy. But this was to just know that even if we have loss of one copy, we do see a decrease in polymerase 2A, which is a co-deleted gene with 17p. So the question is why RNA polymerase 2 is so important? Because among 12 subunits of polymerase 2, it codes for the biggest subunit, and if it is gone, the transcriptional machinery will shut down. And deletion of 17p, as we mentioned earlier, that it includes your co-deletion of polymerase 2A, that makes it more interesting. And that creates a haploinsufficiency. So haploinsufficiency means if the cell has one copy of polymerase 2A, it will be haploinsufficient. That means if you delete that polymerase 2A, the cell will be completely lethal. So therefore, we rationalize that 17p deletion could reduce polar 2A expression and it could enhance the sensitivity to alpha-amanitin. So I'll come to that later. Alpha-amanitin is a poison which is formed from your alpha-amanitin mushroom, and which is a protein specific inhibitor for RNA polymerase 2. And other rationale we use BCMA. BCMA is nothing but B cell maturation antigen. And the best part I like about this multiple myeloma project is BCMA specificity, because this is the only tumor type where I can see that BCMA is expressed only on the disease cells, not on the normal cells. So if you go from maturation from bone marrow to long-lived multiple myeloma, if you see B cells, transitional state, naive cells, or plasmablasts, they don't have any BCMA expression. This BCMA expression is only when the cells are diseased, that is a multiple myeloma. So if you target BCMA, you are targeting only the cell which is diseased. The healthy cells are not targeted. So there is no collateral damage. So now the question is, when we have 17p deletion, we have less polymerase 2A. So it's very obvious that we should check whether the BCMA level goes down or up. So we should not see that there is a low BCMA, so we cannot target it. So we see that there is no difference in BCMA, however, the polymerase 2A significantly goes down. So the present era is of targeted therapy. So this is just an example to show that if you have a toxic payload, if it is not maneuvered properly, there is a lot of collateral damage. So there is kind of missiles which are formed and which are targeted, which can go into a particular bunker and they can destroy the only bunker, not having a collateral damage. So similarly, we need to identify right target, and there has to be a more precision when we have a toxic payload, and there should not be any collateral damage. So death cap mushroom was anciently used for killing people very secretly, because if you keep them with death cap mushroom, they will die within a day or two. It's not a spontaneous death. So metin acts as a toxic payload here in our system, which kills dividing and quiescent cells by inhibiting RNA polymerase 2. Why I mention quiescent cells? Because cancer stem cells are more quiescent in nature, so it can take care of the cancer stem cells as well. However, it's not proven yet. And it will be active against proteasome inhibitor resistant cells. So the proteasome inhibition, getting resistant to it, is another hurdle in terms of treatment of multiple myeloma patients. So if we talk about the payload, amanitin is a payload, and BCMA is for the precision, because we know that BCMA is expressed only on multiple myeloma cells, not on the B cells or the other plasma cells. And myeloma cells will be our target. So these are the BCMA surface receptors, and when we have this antibody, which is specific against BCMA, and it is carrying a payload which is alpha-amanitin, which is a toxin, it binds to the BCMA, gets internalized, and it binds to RNA polymerase 2, because once it is internalized, it is an endosome mediator, and that breaks the endosome membrane, and it goes out and binds to the RNA polymerase 2. And then it shuts down all transcriptional machinery and eventually leading to cell death. So we try to look into the CCNE database, which is maintained by Broad Institute, to see what is the copy number expression of BCMA and mRNA expression of BCMA across different tumor types. So what we found that the multiple myeloma was the leading in front of copy number expression of BCMA, and also the mRNA expression of BCMA in multiple myeloma. However, there are other tumor types like Burkitt lymphoma and B-cell lymphoma, which has level of BCMA. So we are targeting those different tumor types as well with this alpha-amanitin conjugated antibody. So the current status for BCMA as a target is huge and it's very hot in the market. And recently GSK has made an antibody drug conjugate which is BCMA specific, and it has already entered in a phase 2 clinical trial. But when we compare our antibody drug conjugate, which was made by Heidelberg Pharma, I forgot to mention that. So our antibody drug conjugate is water soluble, and the half-life of our antibody drug conjugate is 12 days. However, the GSK antibody what they have made, which is in phase 2 clinical trial, their half-life is only 6 days and it's not water soluble. So I assume that the antibody we have with the drug load that will be more toxic and efficient as compared to this one as well. And there are BCMA T cell bispecific antibodies which can target BCMA, and at the same time they can activate the T cells to kill your myeloma cells. And there are preclinical validation for BCMA as a T cell immunotherapy, and people are using BCMA CAR T cells therapy as well to have their own immune system against your BCMA positive cells, not the BCMA negative cells. And I just do quickly for the BCMA study, because if you wanted to know whether all myeloma cell line we have in culture, they have high BCMA or low BCMA, what is the intensity of BCMA on the surface. So we screen myeloma cells, we screen Hodgkin's lymphoma, follicular lymphoma, diffuse B-cell lymphoma. So these are the different types of lymphomas which showed higher expression of BCMA on the CCNA database. So we try to check these cell lines as well. So we tested almost 20 cell lines for each, and these cell lines for Hodgkin's and follicular lymphoma showed positivity for BCMA. And this data was acquired through FACS, where we pass our cells for antibody mediated positivity for BCMA. And then the whole point was to see whether this HDP-101 is the name of the molecule, which is antibody drug conjugate with alpha-amanitin, to see whether it is more efficacious in multiple myeloma cell line as compared with the normal stroma or any other cells which are BCMA negative. So this stroma cell is a BCMA negative, and all these cell lines are BCMA positive, which is myeloma cell line. And you can see a clear-cut dose-dependent decrease. So as you increase the concentration of drug molecule, there is a significant decrease in your cell viability. That means more the drug, and in a similar correlation, you see a cell viability goes significantly down. And this was done with the cell line, and this was with the patient sample. So once we have a fresh bone marrow aspirate from the patient, we right away process the sample and treat them with HDP-101 to see whether we do see any similarity between cell line versus the patient sample. Because many times the data is not reproducible from cell line to the patient sample. But fortunately, we do see a significant result in terms of patient sample as well. And the best part of this drug was the concentration. The concentration is effective in picomolar concentration, not even the nanomolar concentration. Now, the idea was to test the efficacy in terms of in vitro settings. So we wanted to test how this 17p deletion is more efficacious as compared with non-17p deleted. So we tried to develop an isogenic system. Isogenic system means the same cell having a wild type p53 and wild type 17p, and the same cell having a deletion of 17p to reciprocate the same situation. So what we did, we generated a P53 knockout cells with CRISPR-Cas genome editing. So we edited the part of P53 gene where it was located. We cut out those genes from there, and we made a stable knockdown of polymerase 2
So, we wanted to filter it for Homo sapiens. Sorry. I was wondering, I just had a bit of species snobbery. I thought we should be able to talk. Fruit flies are much more important than others. And we're going to look at an RNA assay, and we want to look at an array assay. And as I said, we'll choose a reasonable one, but you don't always have to choose. Then we'll choose an affiliate sheet 133 plus 2. So I can filter by that. HA 133 plus 2. So that's filtered out where all of those conditions are met. You can see it's got prostate cancer, it's organism Homo sapiens, transcription profiling by array, although interestingly one here has also got gypsy data. And those arrays, if we look into those files, I'm going to sort by assays by clicking on that. And this 17951 is, I believe, the dependent one data set. Okay. So has everybody managed to follow so far? Okay. Okay, so I want to next be able to download the sample data relationship file. If I click on it, it comes up with this. But if I right-click on it, I can save that linked file or download it. Click on a Mac, it says download linked file. On the PC, it says save linked file. So I now go to my downloads. I have this, and that download I can look at in my Finder. So here is my, I'm sorry I can't zoom in more, but it's my SRDF file, and I'm going to open it with Excel.
So is it the same file which we used for doing that?
Yes, yeah, but it really doesn't have to be, but I just want to go through the same one for consistency. So I've downloaded the SRDF file into Excel. Okay. Everybody managed to do that, or anybody struggling? If anybody's finished, if you could help anybody who's stuck. So I right-clicked onto the SDRF file, clicked download file, or on a PC, save file as. Yeah, the reference here is GEOD 17951. Now, is anybody ready for me to move on? Yes? Does anybody not want me to move on?
Yeah, problem? Are you done?
So this file is quite difficult to read in its current form, because the columns are not wide enough for the field, the text in the column. So what we can do to correct that is if we move over this bit and click on it, so that this box in the top left, and then we can move our cursor over the boundary between the two cells, see how the cursor changes. Yeah? And then I double click. It automatically sets the width to the content of the cells. Okay. Are people managing to do that? Yeah, good. Now, this column, A, is our source name. What does that refer to? Sorry? Patient ID or sample ID. Yeah. So that's the most important thing to start with. Okay? So we want to keep that in. Yeah. So the organism we know, or the fact it's a whole organism, we can get rid of the fact it's Homo sapiens, get rid of the fact it's human, sorry, the fact that it is EFO, it's matter suburban. You can get rid of the atomic accession number. Now this one becomes a bit more important.
So what is whole organism here reply?
Well, I would say it's a bit of a misnomer because it's saying it's a whole organism that's been sequenced. But it's not, because it's a sample from a prostate. So, it makes sense. Sorry, did you have a question? So, the description we want to keep. Because that's got valuable information in it. It's got patient number, patient ID number, and it's got the percentage of tumour, which is the thing we want to look at. We'll keep that in for now. And I would say then, everything else we can clear. So this file, or this part, is separated. So just clear everything else apart from the description and the source name. Is everybody okay with that? Good. Right, so this field here, you can see has a number of characters in it. You can see it's got a comma in it, another comma, a point, colon, a semicolon, backslash, colon, sorry, semicolon, semicolon, bracket, colon, T, colon. Now, in Excel, there's a function called text to columns. And I can chop up this string, so that's what we call in mathematical terms a string, based upon certain separators. So I can pass it. So I could use a comma and pull the patient sample out. I could use a colon and pull the ID out. I could use colon here and pull the percentage of tumour out. So I want to chop this string up into usable bits. Now, things you might have heard of Python and Perl. You can do similar sorts of things by passing strings in that as well. So, to do it in Excel, I'm going to go to Data. And please follow along with me if you can. And then I'm going to click on Text to Columns. Okay. That's under Data. And I can choose here either delimited or fixed width. I'm going to use delimited because sometimes the string components can be of different lengths. So that's more accurate. Okay?
Afterwards, going to data.
So it's data, text to columns. And then you select delimited. I click next. And now I can choose from one of a few previously defined delimiters, such as tab, semicolon, comma, space. And some files have these delimiters, so a CSV is a comma delimited file. A text file is usually tab delimited. Some people use semicolons and spaces in certain applications. But I want to choose my own here. And the one I'm going to choose is a colon. And I can preview that there. You can see it's putting in vertical lines. I'm moving around, where it's splitting that data up. Okay, so you can also set things which define text and separate out text. So I've used a colon as the other delimiter. I click next and finish. You can see what it's done is it's chopped it a patient ID here, so we can delete that. Here it's got the patient ID, semicolon, sample type, key tumor biopsy. I can delete that. The next column says percentage of tumor. Nothing in meaningful, they're all identical. I can delete that. And now I have numbers, which is the percentage of tumor.
The 3 columns have been deleted, like KC.
Yes, yeah. And then to be honest, they're just really rubbish text that we don't need. Because ultimately, all we want is the source name and the percentage tumor. So has everybody kept up so far? Yeah? Good. Now, the next step is I want to start getting this into a format where we can start to do some analysis. And the problem with Excel is it doesn't cope with wide spreadsheets, but it copes with long, thin spreadsheets. So if we were to paste in our transcriptomic data, it would crop it off at the end. So if we've got a manipulation in Excel, we need to transpose it. So I have the source name and I have the tumor percentage. And rather than those being essentially organized in rows, we want to turn them over so they're organized in columns. So to do that, if I do Control or in a Mac as well, Command, Control Shift down, Control Shift right, that just selects the numbers. Then I want to copy that, and I'll click here, and I want to transpose it. So to do that, I've copied it, so I click on Paste Special, Values, Transpose. Yep. So to get only the numbers, I select the top left-hand cell, and then I do Control Shift down, and keeping it held, Control Shift held down, and Control Shift down right. So what that does is just detects the point where there's cells with text in them, and so Control Shift, hold them down, and then press down, and then Control Shift, hold them down, and press right.
So after selecting this,
So this will select just the columns.
then what do we do?
So to transpose, we transpose, I literally click copy or Control C, and then I click on the cell up here. I right click, paste special, and I click values just in case there's any formula in there. And what that does effectively is puts all my cases horizontally. So I'm going to delete those bits out now, so I don't need them anymore. And I'm going to format the width of my columns because they've gone all over the place again. So now I just select the text, select everything here, and then just double click on the boundary between. So at this point, we now have a starting skeleton of a file that allows us to pull other files in and start to build up a transcription of a data set. Has everybody got that? Everybody happy?
Yes.
Good. So, the next step is to now get some molecular data. To now get some gene expression data, in fact. So here, now I go back to Safari, or Chrome, or whatever you use, and I want to download the process data. If you want to download the cell files, or the raw data, and process it yourself, then you have to have some tool to do the normalization. But you can download the raw data and normalize it in your own way if you want. But the speed here, and also because I don't have that software installed on here, I'm just going to download the process data. So the process data is stored here in the process.zip file. So if I double click on that, it downloads it. I could also, in effect, right click and download, save download file as. Okay, so is everybody with that so far? You're downloading a zipped file. So a zipped file is a compressed file that's a bundle of data, and then we extract that out. I'm going to go to downloads. You can see here it's taking a little bit of time to download that.
Sorry, so after we have translated.
Yeah. And just leave it as it is at that point. Go back to your internet browser, the Red Express site, which should still be there.
Right,
Then we download one zip file. So in downloading the processed file, you should have some sort of download bar showing you the progress of that download. And it'll be called egon951.process.1.zip.
So what we did till now was processing the raw file, and this is also processed.
This is already processed for you. Yeah. If you read into the protocol here, So mine's now downloaded. Click on that, and you can see here I have all the files with the GSM codes. It's the gene expression profile as a text file for the file name, say the file name. So once your zip is downloaded, open it up and see if you can find these files. Yeah, I think we might be making this download. And you clicked on it, you should have a file list that looks a bit like this. Operating system. Have now the format of relatively still. So the Miami standard is the minimum amount of full microarray. So in the standard, should be recorded through most of these pages like this. Sometimes things in the view format, but if you manipulate and tell, it's quite easy to start to get the. So the term is called data wrangling. Question. It's kind of a standard for what sort of things are reported on this. So it's not the amount of data, it's just the amount of people. So it has to have the array design file, it has to have the identifiers. Right. Now this is downloaded, the next step is to open up, open a file up. So right click, open with Excel. If you're on a Windows machine, you can drag and drop it into Excel, or you can run it on that path. And the file looks like this. So 2 columns. One column is the ID reference, so that's the probe ID, and another column is the level of expression. And this has 2 columns and 54,676 rows. That's why Excel won't cope if you do it. I'll transpose it into Excel and show you what happens. So if I'm just going to go to this stage, actually I'll keep it in. I'm going to click on A, I'm going to right click, and I'm going to say paste special, values, transpose. Show you what happens. So I've got an error. It says you can't paste this here because the copy area and paste area aren't the same size. Select just one cell in the paste area or an area that's the same size and try pasting again. Well, I have selected one cell. What it's actually saying is it can't paste it because it's too much information for itself to go. Whereas if I click here and just click paste, I now have the gene reference ID, and I now have 449147, 449147. So I now, for that case, I have the expression profile pasted in. So if I zoom out, you'll see what that looks like. Yeah, so you've got a long thin column, and then you've got the row of cases going across the top. Zoom in slowly. Okay, so if you all have a go at doing that, if you can, if it's downloaded. So just to show you what I did, I opened this file with the name GSM 449147. That's the array file for the case GSM 449147. So by pasting that in, I now have the gene names in there, and I have the expression values for that case in the data. People managing to do that.
How do we do this for 153 others? Because there are so many of them.
Yeah, it would take some time to do it manually. If you are able to do it with R, you could probably automate it. If you're able to write macros in Excel, so you can record macros quite easily by recording a macro, doing what you need to do, and then automating it that way. So with just one keystroke, you might be able to repeat the whole process very quickly. You could do it in Perl or Python. So all of those will allow you to do it relatively quickly. I'm going to have a look because I have an R script somewhere, not on this machine, but on that machine, just to see if I can. I'm just hopping onto a machine in Nottingham. Okay, sorry, I can't seem to find it on this machine. So, but essentially, it's about 3 lines of code in R. The workflow is to set up a flex grid, to specify the working directory, to point at the file name and open the file, and then so you start essentially with the reference number of the file, open that, paste it in, then close the file, the first file down, open the next one, take the data out, paste it in, and it just repeats that, working through the directory from the beginning file to the end file. So essentially, you put it in a loop that looks at the directory structure and asks, is it at the beginning of the directory, at the end of the directory, and if it's finished, then stop. So how many people have managed to get at least their first expression array profile pasted in? Everybody managing to do it? Yeah? So the next step is to paste the next one in and just keep pasting them in as you go along. You know, it could be automated, and it would be a good exercise to start thinking about doing that with a macro or with R. But once you have it completed, then you have the file which was circulated to you yesterday. In this case. So we took that process to its end point, we would end up with this data set, which is called copy of final data cross data for class. And we have the expression array down the side, patient codes along the top, and then whether there was a presence or absence of phosphate cancer tissue in the sample. So that's, I'll zoom in. That's what that would look like when you finish. Okay? So, for the next step, if you could open up that file, you should have all been given a copy through WhatsApp. Does everybody have that file? Yes. Right. So, the next step is to go through the regression analysis. Are you familiar with that? Do you want me to go through that? Yeah? You want me to go through the regression analysis? No, you're okay doing that, fine. Okay. So the next step is to do something which allows us to get a list of genes. So I'm just going to quickly do a regression because I need to get a list of genes. So this gives us a list of genes. Based upon the standard residual. Sorry?
Cancer 2 versus control 1.
Yeah, so cancer will be on the y-axis, control on the x-axis. So that any expression, if it's above the line, will be upregulated with respect to cancer. So I'm going to sort this by the standard residual. And if you remember, I said I wanted to take anything with a value greater than 6. But if I take anything with a greater value, a value greater than plus 6, less than minus 6, I can delete those out. And okay, I now have a set of genes. So what I did is I've sorted by, I've pasted the gene names in, and besides those, pasted the standard residual. I've then sorted those by the standard residual from smallest to largest. And then because of multiple testing, I can't just use 2, I want to use 6 as the threshold. So I've selected everything with a standard residual of greater than minus 6 and less than plus 6. And then I delete those cells out. So in this case, it's just, I'm ending up with just an enriched set of significant probes. Obviously, if you do it as a nested residual, then you will have more confidence in the results.
So essentially, they're selecting anything putting a threshold at plus 6 and minus 6. Anything above plus 6 and below minus 6.
Yes. Now, obviously, if you do multiple comparisons and average that standard residual, then you have got a way of a bit more of a robust measure. This is just one pair of comparisons. Okay, so once I've got the list of Affymetrix chip IDs, these ones, how can I find the gene names? How do I convert gene and opcode? Spot on. So we're going to paste those names into gene and opcode, and we want to get an identity. So So what I can do is, from gene at home, I can copy this and paste the sheet here. Oops, I don't want to paste it like that. I can edit text. So we want to paste special as Unicode text. And then all I want is gene name and code. If I select this, so lots of text here. If I sort by column H, now have the gene names and for each of the probes. So the process was take the Affymetrix codes, paste them into GeneAnal Home, copy the table from GeneAnal Home, paste that into Excel as Unicode text. And then by sorting by effectively column H, we can get the gene names. If we sort the residuals here as well, A to Z, those now, in theory at least, should align. 152.463 is not there. Hold on, that's why. We can sort these ones by yeah. So now these should match up. I have the gene names here. So from my result, I'm also going to show you I've now got a situation where I've got my gene code, I've got my standard residual, I've got my short gene name, and I've got my long gene name. So do you see how I've done that? Fields were drawn from GeneAnal Home, and then by sorting by the GeneAnal Home result and sorting by my residual result based on the gene name, the two align together.
Yeah, it's ready.
Yeah, okay. So on that note, before we go on to ontologies, we can stop for dinner.
And then by 50.
So we can go for dinner now, and we can return back by 8:50. 50.
It's 8:20.
Actually, I put them in private mode. I have not edit because it take like 4 or 5 hours
For download.
to download. Yeah. Yes.
You will already see.
Yes, sir.
After all of those were
Please move out for dinner. Dinner is already there. And those who have brought the cup for coffee or something, please crash it in the dustbin.
फोटोग्राफर के निकलेगा भी आगे और कुछ भी नंदी आपको भी
ऐश्वर्या चल ना
क्या काम है फोटो खींचना है हाँ फोटोग्राफर बना के सुखाने का लंच देगा बहुत बड़ी मेहरबानी चल नहीं तुम्हें फोटोग्राफर
वो मैम का है चलो चलते हैं क्या बॉक्स है
भाई बड़ी मैं सीरियस ले लिया ये बात को नहीं मैंने भी ले लिया मार्केट में लाइन के लिए आर्थिक बनाना पड़ा था आपको नहीं आपका बड़ा था मैं सब में ऐसा रखना ऐसा निकालना सालनी का भी बड़ा
मेरा कौन ले रहे
Yes, we'll carry on. What I want to do now is select everything. And then I'll switch.
So, now you see it's actually these genes, everything you know, you can delete those. Yeah. Okay. Yeah.
Literature to build up a library. So this is the basis of things like Ingenuity pathway analysis, Metacore, HEAD, to some extent, and string. And these ontologies are constantly updated and it's ever-moving information. And you have to be careful there because if you access, for example, string on the 1st of March 2019 with your 5 molecules, and then you access string on the 20th of May 2019, in all likelihood you will get very different results because the ontologies, the results, the data behind the string system has been updated. So if you reference string, it's important to state the date at which you applied it. Otherwise, people won't be able to reproduce your results, and that's the limitation. So examples of databases and tools. So Amigo that's the URL gene ontology.org. Goa, UEBI, Gorilla, they're wonderful names, these. I went to Goa to see a gorilla with my Amigo. And we encountered a panther. So these are, if we look at panther, we can start to put terms into this. But yeah, please have a look at other databases. So this is Panther. And I want to go to Panther tools and gene list analysis. So Panther tools, gene list analysis. I'll go back. So I'm going to Panther tools there. Yep. Gene list analysis. So in here, I can paste a list of genes. Now, it just so happens, from here, I have a list of genes from my prostate cancer study. Copy those, paste them in, select an organism, and submit. So it then comes up with all the hits for those gene names. And if I click on all, it'll select all of them, but we might sometimes have to have a quick look, like make sure that we've not multiples. But so I've now clicked all, and what I want to do is the molecular function pie chart. So we can see of our 30 identified genes, we have a number of 9 of them, 30% of them are catalytic, 26.7% of them are to do with binding. We have 2 which are transcription factors. We have 2 molecular transducers, and we have 2 molecular function regulators. So you can see we've immediately gone from 30 genes to seeing that there's a lot of catalytic activity in this list, and there's a lot of binding activity in this list. Now remember, this list defines healthy from cancer. We can also look at the biological process. Oh, we've crashed cancer. So we can look at molecular process, but some of you might be able to, but some of you might not. I'm afraid that I think under this broad IP address, there are too many people logged on. But essentially, you can produce, the message is you can produce these pie charts that allow you to explore what the frequency of different functionalities is. And you can look at molecular function, you look at biological processes, you can look at even known pathways. It'll give you that breakdown. Okay? So, we have another approach which is available to us, and these are curated databases. So to some extent, the ontologies are a form of curated database, but other things can be added, more detail can be added. So these are often based on the text mining of literature. So they will search through for associations between things. And they will look for key phrases. And they then score an interaction based upon the number of studies finding the interaction and the number of times the term is mentioned. And different linkages can represent different processes. But you need to check and validate this because sometimes things are missed. Also, ontologies are not always complete. So this is not the gold standard. It's not the only way to look at it because there is missing information. So for example, we used to have a license for ingenuity pathway analysis, and we wanted to look at the glucosyl transferase and glutathione reductase pathways. And we put in glutathione and glutathione reductase, and there was no link between the two. And that's because it was probably published so many years ago that the text mining searchers didn't pick it up in the literature. So sometimes in these systems, the obvious doesn't appear, which is a problem in my view. So Ingenuity was released in 2003. The web page is there. This is a knowledge base, which is based on text mining, and it has chemical, biological, and functional interactions. It's created from millions of relationships from proteins, genes, complexes, cells, tissues, drugs, and diseases, all derived from the literature. So it has a knowledge base, and these are the sources of that. So it looks at textbooks, it looks at peer-reviewed journals, it looks at RefSeq information, it looks at GWAS databases, ontologies, it looks at K, protein-protein interactions, microRNA, mRNA target interactions. All of those are used to define these pathways. don't think I'm going to go through that at this point. So in Ingenuity, you put a search term in a box, you can enter the accession number, the short gene or the long gene name, and you used to be able to access it for free from here. I'm just going to try it. I might have closed the little down. Okay, yes, so that kind of worked. So it brought me to a web page called Gene Explorer. And this is a tool which Sigma Aldrich ultimately want you to use because you're going to buy the reagents and you find something of interest. But you can put a gene of interest in, and then it'll identify products, but it also, certainly also used to identify gene-gene interactions. And these are the sort of outputs you get. So these symbols here identify different functionalities. The dotted lines indicate associations between things. The advantage of of Ingenuity, it's got a broad coverage and lots of tools and features and functions. And disadvantage is how it's expensive. And you can't always make new discoveries because often, because it's literature based, if you find it, then somebody already has already published it. So it's around that. It's expensive because it's around about $6,000 a year for a license, single user license. A free version with slightly less functionality is K pathways. So K is essentially mapping molecular databases to genomics, and essentially you drop your markers into the pathway search, and it will highlight on a known pathway where your molecules fit. In my view, this is much more robust because it's a defined framework. And we can look at K. So I'm going to paste into here, see if it works. So I'm searching the pathway. So to come up with these pathways, so let's come up with a good glycolysis, glucogenesis. Let's come up with biosynthesis. So I went into search pathways. Yeah, that's right. That one. Trying to tell about the pathway. So the principle is at least to drop the genes in and identifies which pathways they are highlighted in the system. I don't think there's a specific strong hits, and it looks like I have hit quite complex. Yeah. I've shown, yeah. So it will highlight components on this. So timing metabolism, for example, it would highlight certain nodes on that pathway. The cake is robust, well characterized, it's free. But again, there's bits missing, hard to make discoveries, and it's hard to make into pathway links. The next one to look at is string. String looks at protein-protein interactions. So now. Hello.
So when we search for pathways in KEK, with every pathway there are two values that come up. One is the p-value that comes up, and there is another which is Benjamin Hochberg corrected value.
Yes.
So what is the significance of these two?
So the p-value tells you for a given pathway, effectively the probability that you've got coverage on that pathway of the markers that equal to. The Benjamin-Hochberg part is a correction for multiple testing. So you can imagine if you have more, if you have say 50 or 100 identities, and you're doing effectively 50 to 100 tests. Those 50 to 100 tests, in order to deal with multiple testing, you have to correct. That's what the Benjamin-Hochberg bit does.
What number of G's that we paste there? What correction is used?
Yeah, so it's based on the number of genes pasted in and the number of genes that are being compared to. The string, this is, oh, it's not string. This is string. We can search and paste our proteins into there. Search. I think I need to put them into Word first. So I go to multiple proteins here, and then search for those. String allows me to search a list. Now thinking about it.
So we have actually pasted the gene names over here that we got.
Yeah, but the gene names are short gene names which will go for given proteins, so that you can use them between. Yeah. And you can see here it's come up. I want to look at Homo sapiens. Find a number of them. Sometimes it has multiple orthologs of the same thing. So, for example, MMP9 has come up with TIMP1, which is associated with it. So, if I click continue here, I now have my string network of just the features I've found. So you can see immediately I've got MMP9, CCL18, MMP12. So these are forming associations. There are no links between these things. And these are the markers that I found. So I can do various things with this. Click, and I can get identities where the gene linkages or protein linkages have come from. And String makes these linkages from the literature. It makes them from energetic potential interaction between two proteins. It has evidence from things like these two hybrid systems, and has evidence from hypothetical finding. So all of these are encoded, and if you look on the diagram here, you can see here, for example, there are multiple lines. So that's a strong link, because that interaction is indicated in all of those different conditions. Other things, I can grow the pathway. So if I click here on more, it starts to add more linkages in. So it adds new nodes in, it adds new features in to try and link everything up. You can see now just by adding a few things in, I've actually added to most of our things here. So we were looking at prostate cancer versus healthy. We've got a number of things coming in. We've got, for example, now Jun B, FOS, MMP9 coming in. We've got SENPA, SENPIN, SEP55. The only things that are kind of isolated are HLA, STAT1, LIX1, now LAMA1 and SERPIN11. But you can see we're now starting to find associations between these. And this is going more from just a list of genes to a systems biology approach. And we can interrogate this pathway by clicking on these links. We can get the papers that have cite these links as well. So it's getting relatively powerful. The only thing is you can export it, and you can export it to various forms, but I would say keep a date, a note of the date when you interrogated this. This changes all the time. So that's string. Any questions so far? So this is free, contains information from multiple sources. It's been developed by a consortium of a number of institutions. Data is weighted, integrated, results of various computational predictions can be inspected, and these are propagated to proteins and other organisms. It's a plugin for Cytoscape, so I'll cover Cytoscape tomorrow. But Cytoscape is a way of visualizing, mapping them out, and creating networks. And you can access string through a programming interface, an API. So you can actually encode it into scripts. If you encode the right URL into that script, you can make a call to a string, get information, and write that result out. These are the content components of string. This is the old data entry. You can see it's come up with a number of hits here for our strategies, and you can see here the interactions that are arriving. The advantages are it's comprehensive, it's free, it's interactive, you can dynamically grow networks. It's still based on a curated database though, so it's still piecing bits of information together. It's not a holistic analysis. So other things we can look at. We can look at gene cards, the Human Protein Atlas, Cosmic. I would also say look at Gene Mania. And there's a whole load, a raft of things that you can look at for ontologies. If you speak to any biotician, you can probably find as many different ontological databases as you can find bioinformaticians. So that pretty much covers the ontological side. What I would say is, while I'm here now, have a go at looking at some of these databases. Put some gene names in, have a play with the result. Because the best way to learn these things is to play with them, to have a go with them. Google Gene Mania, have a look at Gene Mania and see what comes up with that. Because that's another gene-based interaction analysis tool. Have a look at K, have a look at Go, have a look at Amigo. And just see what you find. What I'm doing here really is giving you a basic list of things to look at. And the things that I've worked quite well. Obviously, you might find your own tools. But these online tools are very powerful. Now, one of the limitations of these curated databases is they are based on experiments which are very tightly controlled. So what happens is often people will take molecule A and molecule B, put them in a test tube together and find that they bind or they react or they change the functionality. But they're pairwise comparisons. Whereas in biology, it's a bit more like a web of interactions. In a pathway, if you change this, then it's going to create a cascade through that pathway. They're going to be upstream, downstream effects. There are things suppressing, inhibiting, blocking. And because of the nature of these, they don't give a full picture. And also, they're very general. They're not specific to a given type or group of cases. It's a bit like creating a Frankenstein's monster. You don't have the real human, you have bits of the human, and it's sort of funky. By stitching these together.
But many times you do the list of the proteins, and sometimes in the interaction of in that interaction network, the many proteins don't appear in that interaction. So, but those proteins having significant fold chain or something, how to incorporate that, or can we incorporate it manually in that interaction network?
There's no, in these databases, because they're literally curated, they're only looking for the gene names. They're not looking for the magnitude of expression. If you could incorporate some of that into, if you exported the pathway from string state into cytoscape. If you export the pathway into cytoscape, then you can get differentially weighted lines, and you could weight those lines by association, correlation, covariance, and you can change the size of the nodes based upon the amount of expression. So the algorithm I'll show you tomorrow, which is the network inference algorithm, what we do with that is we change the size of nodes, we shrink and grow the nodes based on the level of expression, and we shrink and grow the edges, the lines between the nodes, based upon the magnitude of interaction. So that gives us a way of visualizing the change, fold change, magnitude of and how interactions occur. So cytoscape is a good tool to do that. And that's also free. I like free things. In fact, I'm going to go to the Cytoscape website and show you. It's in time. The only problem I have is my laptop mic actually. So, in fact, I downloaded Cytoscape earlier.
What is the requirement of the machine classes like that? Software is very heavy.
Cytoscape isn't too heavy. It's 20 meg. So, if I go here and type Cytoscape, this is the current version, 3.7.1. I'm going to download it from Mac. It's taking quite a time. But I was use it tomorrow. Important. How to set nodes, edges, how you visualize things, improve networks, how you can link out things. It's a useful tool for visualization. Does anybody have any questions? So to recap at this point, we've covered the approaches used to download, format, and manipulate data from ArrayExpress. We've covered power analysis, experimental design, randomization, blinded trials, and power analysis. And we've covered ontological tools, databases, where we can search for information. We've we've also covered how we can get gene identifiers from array OIDs. And one of the extensions of that is to look into a website called GeneCards as well, that has a lot of information about genes, their functionalities, what they do. So I think that's covered everything for today. For me, we have some more quiz questions for you to answer.
So please log in. And please write complete registration ID, not just the last. So anybody else is left? So please log in. So now everybody is logged in. Okay, so we are starting now. So now we are starting. So now let's see the final scores. Who is 44? Samika, congratulations. 45.
Oh, okay.
Congratulations, Raj.
Very good. Last 2 days. Yes.
In 41. Congratulations. What's your
You can take it over and then add some.
All right, everyone. So tomorrow again we'll have the same schedule. We'll meet in Bioscience Bioengineering department around 3:45 p.m. 3:45 to 4:45 we'll have 1 hour hands-on sessions over there. Then we'll resume back from 5 p.m. onwards and the same schedule in the same body. As you see today, we try to rush it, because of the so many people moving from there to here takes time. So let's make sure that definitely by 3:45 we start. So preferably 3:30 onwards we can be there. And UG students, if you are missing it out, you can request some other time. We can give you a slot to show the demo again. That's it. All right, everyone. Enjoy the good night and come prepare for tomorrow. Bye bye.
Whatever things you Not this shirt I confirm because I have okay, both are so
All right, so we had another day of workshop today, the third day of workshop, where you try to see that how you can utilize various databases and do different type of pathway search. I hope some of the basics which Professor Graham Ball has explained to you will be useful for you to do a variety of ontology searches and try to make sense of your data which you generate from either transcriptomics, genomics, or proteomics, and try to integrate and see the bigger picture which may emerge out from those experiments. Uh, we are trying to have a blend of the lectures as well as the hands-on sessions. You have heard two of the guest lecturers talking today, giving one from the stand life sciences, giving you perspective of the computational power required for big data processing. And secondly, you have seen the applications of how one could work on the problems like cancer and apply some of these approaches to generate some high quality data. So we'll continue the last day of session tomorrow. We'll have some basics of mass spectrometry based proteomics. We will also have various computational workshop hands-on session and some guest lectures to give you the broader perspective. Have a good day. Thank you.